Today we're going to be talking about mutations. And oftentimes when we say that word, we think about like what we see on TV and the movies. But no, we're not talking about mutants. We're not talking about people with superhero powers. We're not talking about somebody that's going to glow green all of a sudden. Okay? That's fanciful. Mutations aren't quite that. Okay, what a mutation is it's just a change in the genetic code. And as you remember, that code can either be DNA or RNA. By changing that code, this is going to possibly change the protein that that gene creates. Okay, now automatically we think, oh, mutations, oh, that's so bad, so bad. Not necessarily. Some can be bad. Some can kind of just be no big deal or no change at all. And some can actually be really beneficial. We'll see more on that later. There are different causes of mutations. We're going to quickly roll through these. Some of them you already learned about in this class. So during transcription, right, changing DNA to mRNA, if that doesn't go quite so well, and if you get an error there, that's a mutation. DNA replication, right? DNA synthesizing itself. If that copying process doesn't go well, remember those proofreading enzymes catch 99% of the errors? Well, that 1%, there you go, mutations. This is going to be something we're learning about later, number 3. But basically, when your body makes sperm and egg cells through a process called meiosis, if that process doesn't go well, that sperm or that egg could have some funky genetic stuff happening. And if those things become babies, then that baby is going to maybe have some funky genetic stuff happening. Okay. Chemicals. Certain chemicals have been known to cause mutations to DNA. Things like if you've heard of a carcinogen, so things found in tobacco products are carcinogens. Certain pesticides back in the 70s, DDT was a big one that they finally outlawed because it was cancer causing. And the idea there is it ends up affecting your DNA. Okay, changes your DNA. Same thing with radiation. That's why being out in the sun too much can give you cancer, because the sun's rays can actually go into your cells and can change the DNA in your nuclei. Okay. Viruses as well. Some viruses, when they infect your body, they actually take their DNA and insert it into your DNA. And now it's in your body forever. In fact, right now, 8% of your DNA is viral. And you inherited most of that from your parents, who inherited from their parents and their parents and their parents and their parents. So humans right now are only 92% human DNA and 8% viral DNA, which is really, really bonkers to think about. And then genetics too. Some things you can inherit from your parents. So certain genes might be known to cause cancer. Those can be inherited. And so then you're more likely to have certain mutations happen if you get those genes. Don't super stress about that. So I mentioned that like meiosis can be a cause of mutation. And so this is what we call a large scale mutation, in which you have an entire chromosome that's affected, either because it's duplicated or it's missing or the whole chromosome was made wrong. Okay. This is going to be a large scale thing because it's going to deal with a vast amount of DNA. And one example of this is Down syndrome. Okay. We're going to be learning more about this in like a week or two. But the idea here is that an typical human has 2 copies of each chromosome. But notice on number 21, there's 3. And having that 1 extra chromosome leads to Down syndrome. Okay, this would be what we call a large-scale mutation because a large chunk of DNA is affected. But what we're going to be focusing on today are small-scale mutations. Instead of like entire huge chromosomes being affected, we're just talking about part of a chromosome is altered. Okay, most of it's right, but part of it's gone wrong somehow. Okay, and so we're going to be zeroing in on that and looking at 2 different kinds of small scale mutations: point mutations and frame shift mutations. A point mutation is also known as a substitution. And so what that means is there's going to be substituting one nucleotide for another. So substituting an A for a T, an A for some other letter. Doesn't have to be a T. An A could become a C. And all of a sudden, that's going to change everything. So sometimes it's better to just see it with an example. So if I have this sentence, the fat cat ate his fig, not his hot bun. Okay. But now the substitution here, the point mutation would be the fat hat ate his fig, not his hot bun. So where is it? Going to be right here, right? Instead of cat, there's an H in there for hat. So most of the sentence stays the same. But this mutation changes the meaning, right? Because how does a hat eat? What? Hats don't eat. Exactly, right? So this causes, obviously, clearly a different message. Okay, so we can have point mutations where just one letter is changed, and these can have a big effect, a little effect, or no effect. So let's take a look at that. And here's an example of no change. It's called a silent mutation. And this is when the mutation ends up making the same amino acid that the original one had. Do you remember last week when we were using our codon charts and how some amino acids had several different codons that would lead to the same amino acid? That's this. So here's an example. We have GAA, codes for glue, and then here's our mutation. Instead of GAA, it becomes GAG, but guess what? Using our chart, that also codes for glue. So we end up with the same amino acid sequence, which means this is going to become the same protein, which means we're going to see no effect at all. That's why it's called silent, because the mutation doesn't end up making any noise because it ends up making the exact same protein. Sometimes we can have an effect where just one amino acid changes. Okay, this is called a missense. So here's an example here. The GAA becomes a GAC. So instead of glue, we get asp, okay? And so notice the rest of it's the same, met, alagli, but the glue becomes asp. And you're like, okay, so what? Well, with proteins, their shape is determined by their amino acid order. And so by just changing out one amino acid, you're probably going to change the shape of this protein. So if this is what it was supposed to look like, maybe now it looks like this. And so depending on what that protein does, that might mean that the protein doesn't work at all, or maybe it means that it just kind of sort of works, or it doesn't work as fast, okay? So we might have some issues with this protein because it wasn't made by the proper sequence of amino acids. The next type of mutation is called a nonsense mutation. And this is when, instead of just like the last one where we made a different amino acid, this one codes for a stop codon way before it's supposed to stop. So this is going to produce a way shorter protein. And that protein might not work because it's too short. It's going to be the wrong size, okay? Here's a nice visual, just kind of showing you all of them side by side to help you wrap your head around it, okay? Remember back when we first started talking about it with small scale mutations, there's two main kinds, point and frame shift. We just talked about point. Remember, point is where, you know, the C in cat becomes an H, possibly. Okay, just one little letter is changed. Well, now we're going to talk about frame shift. Frame shift mutations are going to be different. There's two different kinds, insertion and deletion, and the names tell you what it is. An insertion is when one nucleotide, one letter, an A, T, C, or G, is inserted somewhere. And this is going to change every codon after the insertion. So let's go back to our original sentence. The fat cat ate his fig, not his hot bun. Well, if somehow an I gets inserted here, but all the rest of the letters stay the same, because codons go in triplets, it's going to bump everything off by a letter. And so now it's going to read the phi, taka, tat, ahi, safai, gno, the, show, taboo, right? So, wow, is most of the sentence same? No, everything after the insertion is different. Does it change the meaning of the sentence? Yes. This now like doesn't make sense at all, right? It's complete nonsense now. I'm saying nonsense as like an English word, right? Complete mumbo jumbo, right? Like you don't know what that means because it means nothing. So next is deletion. And so it's going to be like insertion, but the opposite, okay? One letter is going to be deleted, and that's going to shift over our codon triplets, okay? So here we go. Here's our deletion. The fat, what comes after this T should be a C. Uh-oh, there's no C. It just goes straight to that AT. So now our sentence becomes the fat ada te isif ign oth ish atp un. Right? Notice we're one letter short here, whereas here we read one extra letter. So this is going to change every codon after the deletion, which is going to cause this to, again, not make a lick of sense. Okay? So let's look at the effects of frame shift. Remember the effects of a point mutation. We had three different options, silent, missense, and nonsense. With a frame shift, silent is not possible because if you notice, all of these changed. And if these are going to stand for amino acids, it means that we're going to have a whole bunch of amino acids. And so we're never ever going to have a silent mutation. We're never, ever going to have just one go for the other, and that's it. There's going to be huge differences here. And so at least one amino acid is going to get changed. You're not going to have them all be the same. And so a silent effect is not possible from a frame shift. Can we have missense where we get different amino acids produced? Yeah. Basically, like most of them or all of them are going to be different. Everything after that insertion or deletion is going to be different. So this protein is going to maybe work or not really work at all. Okay. What about nonsense? That's the early stop codon. Yep. If you shift around the letters like this, we could totally cause an early stop codon, which could lead to a protein that doesn't work or only kind of works. So we've been talking about this kind of in abstract, but I want to get really specific now with humans. And what are some examples of mutations in humans? Let's talk positive first. Okay, what are some beneficial mutations? Do you know that humans, or actually like most humans, are lactose intolerant? Yeah. And so if you literally in your DNA, one C gets switched with a T, and now you can digest lactose. Hooray! Okay. And so now it's super, super common, right? But there are still people who are lactose intolerant, but what that means is they just don't have the mutation. Okay. So this ended up being a very advantageous mutation for us because, you know, dairy products are now such a large part of our diet. Same thing with color vision. Okay, most animals only see in monochromatic or dichromatic, meaning one or two colors. They can't perceive all the colors we can. But humans, we can see in 3 colors of light, red, blue, and green, and they mix together to form all the rest of the colors. But a couple million years ago, somebody got a mutation that allowed them, in their color vision, that allowed them to see different colors, and now, boom, like this is what humans see now. Now we're also seeing some people with an HIV tolerance. You don't have to worry about it, but there's something in their cells that keeps the HIV virus from infecting their cells. Wouldn't that be a great thing to have, right? Some not so beneficial mutations would be cystic fibrosis. Maybe you've heard about this, but it's a pretty nasty disease. Same thing with Tay-Sachs. These are just single little mutations that have big effects on the body. Color blindness. Okay, some people are colorblind. Now that doesn't mean they can't see any color. It just means some colors look the same. Okay, and that's from a mutation. Some cancers can be traced back to mutations, one or several mutations. One type of mutation that's not really beneficial or negative is blue eyes. Okay, having blue eyes, getting that blue color was just a small genetic change. But of course, having blue eyes doesn't increase your chances of living or decrease your chance of living. It just kind of is a thing that kind of looks nice, right? But it's not good or bad. It's just there. So just in summary, mutations are from typically a change in DNA, but it could be RNA. Okay. And depending on what the change is and where it occurs, it can have big effects, it can have medium effects, it can have no effects. We've got point mutations, we've got frame shift mutations. Hopefully now you've got a little bit better handle on them. So when we go to practice with these in class, you're good to go.